It's streaming.
Yeah.
Oh,
Can I just take
It looks like everyone is here, and I am here, but not the least. And this Monday morning was troublesome for many of us, and I'm living on an island, and there was problem with ferry over today. Never used to be, but today I delayed all of us for 1 hour more than that. But now eventually we can start. And this dissertation, and dissertations are really important part of the scientific life. As you know, science is all about to build models. Model building is what we are doing together when we are discussing science. And always when we are building these models, there are different opinions of how to interpret data, how to trust the data, and how to build the models. And so the scientific discussion is the most important part of this model building, to mix this jigs of pieces together. And this is what is going to be done at the dissertation. And the dissertation today is made by written by Kittichate Visuttijai. I'm not so good in your name, sorry for that, but I hope I spelled it so everyone can recognize it. And on the opposite side of the border is we have faculty opponent Professor Volker Hacke from Institute of Molecular Pharmacology. Welcome to you. We are happy to have you here. And we have also a committee that are going to follow the discussion and add on questions in the end of it. And we have Professor Maria Falkenberg from our own institute here, and Professor Fredrik Stahl from Högskolan i Borås, before working with genomics, I recall.
Yes.
Yeah? And Professor Peter Karlsson from Department of Cell Biology, also at Gothenburg University. And after the discussion is finished and all questions made by the committee, also the audience have the opportunity to ask questions if you have still something to add or something to some question marks to strike them out. So you're most welcome to do that part. So I believe that the opponent is going to start with a small introductory. So welcome.
Good morning, everyone. It's my pleasure to be here. And as the instructions said, I've tried to keep this really simple. I realize, looking at your faces, you cannot possibly be a lay audience because you look so knowledgeable. So I apologize already that this may actually be much of what you already know, but in order to facilitate discussion and perhaps to keep the few of you who may not be too familiar with the topic up to date, I present some of the correct overview of what is known about cell signaling in cancer. That said, I'm not myself a specialist in cell signaling, let alone cancer. We're cell biologists studying the role of phosphoinositides in membrane traffic, and at some point these cross paths with signaling networks. So I'm a lay more so than perhaps you are. So what you're looking at here is an image that I stole from Memorial Sloan Kettering Cancer Center in New York, where you can actually see a tumor cell being targeted by immune cells that actually attack this tumor cell. And this very much outlines the problem much of medicine when it comes to battling cancer. Cancer cells are not only proliferous cells, but they're also cells that try to evade the immune response. And as you can see by their shape and these little tentacles, phylopodia, they actually use to migrate out of the usual environment, the tissues, and they do metastasis in other parts of your body, and this is perhaps what may in many cases be most detrimental to the patients. There are different kinds of cancers. There are so-called carcinomas, which are perhaps the most common types of cancers, and Kitty has been studying two different kinds of those. Leukemias that originate from cells in the blood, lymphomas that are, of course, cancers of lymphocytes, and sarcomas that are more rare but ever so more deadly in many cases. Now, what are the hallmarks of cancer cells? Kitty has alluded to it in the introduction of his thesis, and this is in fact taken from the renewal of a very famous review on the topic by one of the godfathers of cancer biology, Bob Weinberg, who's written this in a relatively recent edition, 5 years ago, published in Cell, where in fact some of the hallmarks of cancer are summarized. And these hallmarks are sustained proliferative signaling. That is, of course, perhaps at the origin of what all cancer cells arise from, because the cells have to go through mitosis in a dysregulated fashion in order to produce more progeny than they normally would do. However, that said, that isn't usually enough to cause a cancer, because we have an immune system that may actually eliminate them. And therefore, a number of other changes, usually more than one. My poor knowledge says at least two, but perhaps more changes occur, that have to do with things like resisting cell death. Normally, cells actually have a control system, apoptosis, that actually would eliminate supernumerous cells, and it does so in development. So we're actually producing a lot of new cells in development, but usually we do not develop cancer, and that is because in development, cells actually have programs that can actually eliminate supernumerous cells, as they do, for example, when you're actually developing your hand. The fingers are developed by elimination of cells from between. Another important issue is tumors, of course, have to sustain growth, and in order to do so, they have to be supplied with the goods that any cell needs in order to proliferate, and therefore induce angiogenesis. And furthermore, they actually show features, usually in later stages, where they actually start to evade tissues, the tissue they arose from, and invade secondary tissues where they form colonies that grow as metastases. So these are some of the things that we'll have to keep in mind when talking about signaling in cancer, and that is about as much as I can tell you on the topic. As I said, many of you maybe may know better than I do. Now, when it comes to the drivers of cancer, there are two important distinctions when it comes to the kinds of genes. And I put this relatively simplistic, so the ones that don't think about it, the ones of you who don't think about it every day, actually may get a feeling for what is meant by this. Now, tumors, well, growth in general, is controlled by and large by two kinds of genes. Genes that, when mutated as gain-of-function mutations, will accelerate growth. These genes are called oncogenes, and one of the sort of famous examples is the RAS oncogene. These are cellular genes. If they're cellular genes, they're called proto-oncogenes. That is, when mutated, they may actually acquire oncogenic properties, which eventually leads to a signaling function irrespective of whether the growth condition is supportive or not. So, by analogy, they act a little bit like a car, where you have an arrested gas pedal. You just keep accelerating, and no matter what you do, you will eventually hit the wall. Now, oncogenes are dominant mutations. So it may actually be good enough to have one mutated RAS that overproduces active RAS protein or overproduces a receptor that will lead to sustained growth. Very different in a genetic sense from these, as far as at least I understand, tumor suppressor genes do exactly the opposite. They normally act as brakes in the system. So the mutation, usually as a loss of function allele, will result in the loss of the brake paddles in our car analogy. And if the brake system is disabled, you will also generate uncontrolled growth and cancer. And famous examples of these are the P53 tumor suppressor, but there are many others that are being identified, and we'll hear one example today from Kitty's work. So, unfortunately, of course, cell biology isn't that simple, right? Not to say that cars generally are simple, but for sure, any eukaryotic cell has many more components, many more different components than any car has, and hence signaling networks that can result in cancer are also complicated because you have intersection of multiple pathways that on the one hand control cell proliferation and growth, and we'll focus on these today. But there are also other pathways, for example, pathways that control motility. Now, motility, as I said, is one of the hallmarks of cancer that many cells acquire secondarily, because when they actually evade the primary tumor to form metastasis, then this increased cell migration and invasiveness has to do with these motility circuits and the dysregulation of these circuits, different cell adhesion processes, and so on and so forth. There are other circuits, viability, for example, in order to evade apoptosis and usual elimination. So there is really a lot going wrong in tumors, and that's perhaps one of the biggest problems you're facing when you're examining cancer cells. And you would probably know way more about this than I do, but if you actually look at cancer versus normal tissues, you will find many, many changes, and it gets very difficult to discern what is primary and what has been secondarily acquired in order to really trace what has been going wrong in the first place. This complexity is also one of the problems when battling cancer. Many therapies against cancer have been developed. These include inhibitors of the signaling responses that drive cell proliferation in the first place, but also many others that have to do with replication, with the question of angiogenesis, metabolism. We all know that tumors essentially run on glycolysis, whereas most other tissues in our body use respiratory growth. So one uses inhibitors of metabolic pathways and so on. So let me zoom in now on a few aspects that I picked more or less arbitrarily because I think they're relevant to the discussion that Kitty will present to us in a few moments, and that's the proliferative circuits. Now, cell growth and proliferation obviously is one of the most tightly controlled aspects of cellular life. We are fairly stable, aren't we? So, as I'm speaking to you, my brain and my nerve cells, which is another topic that we're caring very much about in the lab, in fact, are the very same nerve cells that I was using when I was a first grader and had my first day at school. And then, in fact, used the very same synapses in order to memorize this effect. So, these neurons, very obviously, have not decided to undergo division and make more of them. However, other parts of my body have changed considerably. Some of them have grown, others have aged, unfortunately. Some of them even may have died. But that says we're in a very tight balance. And biology has put a lot of effort into balancing these signals that make up this decision in this tight balance. So you may only need a small twist in the system in order to tip the balance, and therefore it is important, but it's also complicated and challenging to understand and dissect these signals. So let me give you the textbook knowledge before we're moving on to the latest that Kitty will present. Now, growth requires factors that tell a cell, I'm in a good environment. I'm supposed to grow, I'm supposed to survive, maybe I'm supposed to proliferate. In your epithelial cells, this happens a lot. So these growth factors, in fact, are molecules that usually come about as dimers. The sort of famous example of this is epidermal growth factor and its family, which is dimeric molecules bind to receptor tyrosine kinases. One of these kinases, HER2, in fact, is one of the famous oncogenes that, when overactivated, that is constitutively active, can drive tumorigenesis on its own to a certain level. Ligand association of these receptors results in cross-autophosphorylation events, and then the recruitment of signaling factors, of which there are several different branches. And I don't have time to tell you about all of them, but I'll tell you about two. The most conservative, the perhaps most classical pathway that has also received a lot of attention early on is the RAS pathway. And the reason it's received a lot of attention is twofold. One of them is it's been long known that mutations in RAS can also drive tumors. So you have two oncogenes in the same pathway, HER2 and RAS, that on their own are found to be mutated in lots and lots of different tumors. The second reason is this is a pathway that has been studied because of its evolutionary conservation, and fly photoreceptors would actually use RAS signaling for development. And this is where the picture is actually taken from. It's a textbook picture from Albert's textbook, but this is a Drosophilus. It doesn't matter because the principle is exactly the same in mammalian cells, where receptor activation results in recruitment of effector proteins, such as this protein here called GRAB2 in mammals, called DARK in the fly, because the flies can't see if this protein is mutated, because they don't develop the photoreceptors properly. Never mind, this recruits a protein that acts as a RAS GIF called Sus. Son of 7 less is where the name comes from. It's again a fly name. And this will catalyze nucleotide exchange on RAS, which will signal to downstream effectors to control gene expression. And it does so by a kinase cascade that involves active RAS, then another small g protein called RAF that is activated through RAF kinase. And this in turn results through another cascade of kinases in the activation of a kinase called the MAP kinase. And the MAP kinase is a latent transcription factor that, upon phosphorylation, will translocate to the nucleus and activate target genes that you actually need for cells to proliferate. That's a classic pathway. However, and constitutively active RAS is an oncogene, and I said that. However, that's only, unfortunately, a fraction of the story. There are other pathways that also contribute to signaling from these receptors, and I'll ignore two of them, but I'll tell you about one, and that is the PI3 kinase pathway. That is because we ourselves are interested in PI3 kinases, but more importantly, because you have actually, Kitty, started to elaborate on this pathway. Now, this pathway is elicited, again, by the recognition, and that's the sort of common denominator of all these events, of phosphotyrosine residues. Phosphotyrosine residues are recognized by so-called SH2 domains. You've seen one in the GRAB2 protein on the previous slide, right, if you remember. PI3 kinases, and in this case class 1 PI3 kinases, that actually are dimers, also carry an SH2 domain, and this serves to see this activated receptor tyrosine kinase and then recruits the catalytic domain of PI3 kinase to change plasma membrane phospholipid content in a particular fashion. And this kinase, what the kinase really does, it's producing a lipid called PI345P3 from PI45P2. Now, this is perhaps for the lay audience the most complicated slide. What you're looking at here are a minor class of phospholipids called phosphoinositides. They're all derived from this mother lipid that is comprised of a typical glycerophospholipid and an inositol polyalcohol. The inositol, in fact, is in principle a very boring molecule, but it has one interesting property, and that is it can reversibly phosphorylate almost any one of these alcoholic hydroxyl groups at different kinds of positions, resulting in 7 different species of phosphoinositides. Now, what these do is they connect 2 things, 2 important pieces of information that we're all interested in biology: space and time. The molecules are located in different places. PI45P2, the substrate of class 1 PI3 kinase, is the marker for the plasma membrane. So this lipid tells you, I'm at the plasma membrane. Now, the second important thing is time. That is, class 1 PI3 kinase is constitutively inactive, but only when the signal arrives will it be recruited locally to receptors and will phosphorylate 4,5 into PI345P3, which, as you will see in a moment, then activates downstream effector molecules. As I said, signaling is a check and balance system. We need to be certain that signals are actually transient, because otherwise they will drive cells nuts. And in order to do so, the system is counterbalanced by a phosphatase called P10. Now, since we were talking about oncogenes and tumor suppressors, P10 is one of the most important tumor suppressors that are out there, and many tumors, in fact, carry lots of function mutations in P10 that will result in elevation of PIP3 signals that are found, again, as a hallmark of many types of cancers. Now, PIP3 not only can be reversibly phosphorylated and dephosphorylated, it can also be dephosphorylated to the second lipid that we may actually discuss as we're moving along by the so-called SHP or IMPP5 phosphatases to give rise to PI3,4P2. And this results in a certain complexity of the system that we'll probably discuss as we're moving along. So anything that we're talking about here we think is occurring at the plasma membrane. So all of this red box stuff is local at the plasma membrane. And we'll see what that means. So how does the lipid now operate? The lipid in itself is an activator of yet another kinase cascade, completely distinct or partially distinct from the RAS-MAP kinase signaling that centers around a kinase called AKT, or protein kinase B. Now, AKT is important because not only does it trigger responses of cells that have to do with cell shape and migration, but it also controls and is controlled by a complex called the TOR complex. Now, if you actually do research on TOR, you will find that TOR perhaps is one of the most important regulators a cell has. In fact, our body has to control cell growth because it's intersecting at many different places. It gets signals from various kinds of pathways. It integrates nutrients, growth factors, metabolism, cell growth, and so on. So whether or not this is active is one of the key features that any eukaryotic cell will have to decide on. So AKT undergoes phosphorylation through, in fact, 2 different kinds of phosphorylation events that Kitty has also been using as a readout. One of them is the phosphorylation through a complex called mTORC2. This is a mistake, which phosphorylates AKT at one particular site, and this then triggers phosphorylation by a second kinase that results eventually in fully active AKT, which, for example, serves as a signal to overrun apoptosis or any kind of apoptosis production, and it will actually promote cell growth and division. It will promote translation and many other things that we can discuss about. So as a last bit of information, I would like to move from the signaling circuits to cell motility and just leave you with a piece of information that, in fact, these proliferation circuits and the motility circuits are interconnected. That's not fully understood in all details, but the sort of textbook knowledge says that cell signaling not only leads to changes in protein production, translation, replication, and so on, but it also changes cell shape, and it changes cell shape in particular with respect to the cortical actin cytoskeleton, because cells actually activated cells tend to move, and they do so via signals that are triggered by PI3 kinase signaling, but then intersect through various other effectors and pathways that have to do with PI45, P2, also with PIP3, that eventually lead to a remodeling of the actin cytoskeleton. Now, actin is important not only to keep your cells in shape, but also to convey movement in a directed fashion. So, in fact, when growth signals, pro-growth signals, are seen by cells, they usually have some sort of spatial gradient. It's a bit like the children who actually see this little hut that has all the sweets, and you can see the directed movement towards the sweets. The cell essentially does exactly the same. It smells the gradient, it smells the sweets, and then remodels its actin cytoskeleton in order to convey a directed movement that is done by motor proteins of the myosin family in order to bring cells to where it actually wants to go. These myosins come in different flavors, and they generally act by, on the one hand, binding to actin with their so-called motor domains, which you can see here as these little globular heads, then some variable domains of different lengths, and a C-terminal end that allows them to bind to other things, to cargos in particular. So this movement is a movement that has been understood and dissected in structural and biophysical detail, where the myosin head migrates along the actin filament through a sort of a conformational change with a push movement that changes the myosin head. And this will be one topic that Kitty will further elaborate on, because his tumor suppressor, in fact, is a member of the myosin family. So the battle against cancer certainly isn't won. Definitely not if you're a human, maybe if you're a mouse. And so we'll have to keep going with respect to dissecting further tumor suppressors and oncogenes to actually understand the tumor response. I've tried to give you a bit of an integrative overview of what I felt was important to now follow on Kitty's remarks. And with that, I'll be done for now, off the stage, and I think it's Kitty's turn. Thank you.
Good morning, everyone. Good morning, Professor Walker, the opponent, examining committee, also my supervisors and my co-supervisors and everyone here. Today I'm going to present my dissertation under the title Cellular, Molecular and Functional Characterization of Tumor Suppressor Candidates, MYO1C. To begin this, I'm going to make a synopsis of the presentation. I will start with the background, result and discussion, concluding remarks, future perspective, and ending with the significance of the PhD study. Cancer. Cancer is a group of disease characterized by the growth, invasion, and spread of cells from original site to the secondary sites of the body. And since I say that it is a group of disease because it contains more than 100 types of cancers or in different organs of the body. And Professor Walker has already talked about the hallmark of cancer, but I will just saying something a little bit on some of the hallmark. For example, activating cell and metastasis. The normal cells usually stay in one place or in one organ, but cancer cell is differently. They can move, they can act, or they can invade the other type of cells. Also, normal cells, they grow with the external signal, by the external signal, but on the cancer cells, they can sustain or maintain the growth by itself. That's why one of the hallmarks of cancer is the sustaining proliferative signals. And also, normal cells, they are controlled by or destructed by the apoptosis or cells death. But cancer cells can invade this process. And this thesis will emphasize on the tumor suppressor. And tumor suppressor is a gene or a group of genes that regulate cells number and ensure that the cells are healthy. And healthy in this context means that they are behave like normal, act as normal, and has a phenotype as normal as it is. And the product of these genes act as a negative regulator to inhibit cell cycle, promote differentiation, and then trigger apoptosis. There are two types of cancer, and the classical tumor suppressor genes follow Knutson's two-hit model. That means that in the deactivation of this type of genes, you need the deactivation of both allele. And there's another type of gene that is behave like haploinsufficient, and this means that only one allele can trigger the phenotype. The overall of my PhD study, the first one is to identify tumor suppressor candidate in the region of recurrent chromosomal loss. The second, to investigate tumor suppressor phenotype of myosin 1C and potential correlation with the PI3 kinase and ACT signaling, as well as RAS and ERK signaling. Third, to investigate expression pattern of myosin 1C in colorectal cancer. And fourth, to explore genes and pathways through which myo1C can exert its cancer-related phenotypes. And this thesis is based on one publication and 3 manuscripts. So, starting with the first publication, analysis of an independent tumor suppressor locus turmeric 2-TP53 suggested INPP5K and myosin 1C as a novel tumor suppressor genes candidate in this region. The background in this previous work has identified a candidate tumor suppressor region, and this region was commonly deleted, and it is independent and distal to TP53 locus. In this region, it has spanned about 700 kilobase that harboring 19 genes. In this paper, we conducted various methods to identify or to single out the candidate genes. Out of 19 genes, there is 9 genes. I have to go to this. I'm sorry. Out of 19 genes, there is 9 genes that it that its expression has been downregulated in comparison between normal cells and the endometrium cells. And then out of 9 genes, once we divide it into 2 groups, that is a group of cells that has the normal chromosome, and then another group that has the deletion or allelic imbalance. We found out that it's 5 genes that has the downregulation regardless of the deletion. So we pick out that 5 genes to be studied. And after that we do sequencing PCR and there is no mutation. So the cause of the downregulation or deactivation of those genes is not causing by the mutation. And we check the protein expression to check that the expression of mRNA and the protein level are correlated. And the last one, we did the epigenetic study on promoter methylation analysis. And out of that 9 genes, when we compare to without the deletion, it comes to 5 genes. And then the 5 genes we having I'm sorry, I don't have the laser pointer. We have HIC1, RPA, IMPP5K, myosin 1C, and CRK. And then we opt out CRK because CRK is very famous oncogene. And then for the HIC1, we found out that the protein expression is not correlated with it's not downregulated. I'm sorry. For the HIC1, the protein at the protein level, it is not downregulated as an mRNA level. So for 5 genes, we have the 2 genes left, that is IMPP5K and myosin 1C. So the conclusion of paper 1, we have IMPP5K and myosin 1C as a best tumor suppressor candidates. And these 2 genes has overlapping roles in PI3 kinase and ACT signaling. However, there is no earlier report on these 2 genes act as a tumor suppressor. But the families of these 2 genes are reported that act as a tumor suppressor. For example, for IMPP5K is a P10, which is phosphatase enzyme, and then for myosin 1C is myosin 1A, for example. And this thesis will focus on myosin 1C. What is myosin 1C? Myosin 1C, also known as myosin 1 beta or MER2. It is unconventional myosin. Unconventional myosin in this case can be described into 2 things. The first thing is that they have like 1 head, because normal myosin has 2 heads to crawl along the actin. And another reason is that they do not behave like normal myosin in the muscle contraction, but it behaves in a vesicle trafficking. There are 3 isoforms. 2 isoforms we found in the cytoplasm and the nucleus, and the last isoform we found in the nucleus only. And at its tail, it contains a pH domain that allows the binding tightly to the inositol phosphate, such as PI45P2 or PIP2 in this context. And then, like I said, it is unconventional, so it involves in glucose metabolism through mobilizing the glucose transporter 4 or GLUT4 containing vesicle in response of insulin. And in this picture, you can see that myosin carry the vesicle that contain the GLUT4 along the actin to the membrane as a response to the signal of insulin. And also another function of it is jointly to the E-cadherin to maintain the cell adhesion. So we come to the paper 2, which is lower expression of tumor suppressor candidate myosin 1C, stimulate cell proliferation, activate ACT, and suppress cell adhesion. The aim of this paper, we examine the original hypothesis of myosin 1C that act as a tumor suppressor in the relationship to the hallmarks of cancer proposed by Hannahans and Weiberg. In the experiment, we perform gene expression analysis and then cells transfection, and followed by the assays such as proliferation, migration, spreading, and adhesion assays. The last one, we do a protein expression analysis component of PI3 kinase and ACT signaling, as well as LARS and ERK signaling with a myosin 1C overexpression or knockdown. So the result for the first one, we did the expression by performing immunohistochemistry, and we found out that expression of myosin 1C was significantly down-regulated in low differentiated endometrium carcinomas. As you can see from this paper, from this graph, you can see that at the low level, we can see a lot of half of it can be found that myosin 1C was expressing in a very low level in all. And the second result, we found out that overexpression of myosin 1C significantly decreased in cell proliferation. So what we did, we transfect the cells using the construct containing full length of myosin 1C, and what we find is that at the concentration of myosin 1C construct at 0.9 microgram per milliliter, we have the significant lower number of viable cells. And this is by vary of the concentration, and then we use the same concentration and vary by the time. We found out that after we transfect the cells with 0.9 microgram per milliliter, from 48 hours up to 96 hours of our observation, we have the lower viable cells. And once we knock down the myosin 1C using siRNA technology, we increase the cell proliferation. This plot showed that the knockdown using siRNA was successful, and then after 48 hours, up to 96 hours of the observation, once we knock down myosin 1C, you can see from this, this is a positive, +1 means that it transfected with myosin 1C, 24, 48, 72, and 96. And then you can see that from the 24, the protein just starting to decrease. And then we have the result saying that for 48 hours, we have the lower of viable cells. And then when we knock down myosin 1C, we also impair the cell's migration as well. And this one was conducted using the migration assay. So starting with, we press the cells into the well that containing a stopper. And once the cell has been established over 24 hours, we pick up the stopper out, so we create a surface. And then we let them crawl to cross. It's just like wound healing. So after we knock down myosin 1C using the siRNA, we found out that the cells that has a lower myosin 1C can cross the space slower than the one transfected with a scrambled siRNA. The next result, we found out that lower expression of myosin 1C also impair cell spreading and cell adhesion. And this one we conducted with a different amount of siRNA from 1 nanomolar up to 10 nanomolar. And we found out that at 5 nanomolar and 10 nanomolars, we have smaller amount of the cell that can adhere to the surface. And this also means the cell spreading as well. The next result, we found out that once we overexpress myosin 1C, we have the increase of P110 alpha, decrease P10, and as well as it activated form of phosphorylation. So we transfected with a different amount of myosin 1C construct from 0.3 up to 0.9, and then this one is a blood of P110 alpha, and we found an increase of it after normalization. And then the rest is a decrease of P10 and decrease of ACT as well as phosphorylated ACT S473 and then T308. And we knocked down myosin 1C also with the siRNA again, and then we starved the cell overnight to have the to perform a induction later. So what we did is that we grow the cells and then we transfected with the siRNA to knock down myosin 1C, and then we grow it in the medium with our serum. So, and then the day after, I activated the cells using the serum, and we collect the protein from 2 minutes, 5 minutes, and 20 minutes after activation with serum. And what we found out is that we have the faster activation of phosphorylated ACT compared with the cells that transfected with siRNA and the cells that act as a control with a transfection with the scrambled siRNA. However, we cannot find that activation in terms of p-ERG. I've done this because ACT and ERG are signaling that it is like neighbor, and there's one report saying that if we block ACT, things can happen in ERG, and if we block ERG, things can go back to ACT as an example. So this is what the reason behind. So the paper 2, we conclude that negative association between myosin 1C and endometrium tumor, endometrium carcinomas tumor stage. And there's a negative correlation between level of myosin 1C and activation of PI3 kinase and ACT signaling and cell proliferation. And when we knock down myosin 1C, we impair cell migration, cell adhesion, and this could support the invade of cells from cell contact inhibition. This work provides evidence for the involvement of myosin 1C in a contact inhibition and cell proliferation. For the third paper, expression of myosin 1C is down-regulated in primary colorectal cancer tumors. The aim of this paper is to investigate the expression of myosin 1C in the clinical samples and a potential correlation between myosin 1C and expression and activation of PI3 kinase and ACT signaling. And this has been done because the very first result we found in the endometrium cancer, and then we want to check in another type of cancer that is quite in the different organs, but as a child the same type as the carcinomas. So the experiment we perform RT-QP, we perform qPCR Western blot analysis of myosin 1C in a panel of 24 pairs of colorectal cancer and the corresponding normal appearing mucosa. Also, we did a Western blot analysis for the component of PI3 kinase and RAS signaling in those samples as in the colorectal cancer samples. So the result, the qPCR showed lower expression of myosin 1C in the tumors compared to their corresponding mucosa. And this happened when we divide it into according to that stages as well. The second result, the Western blot showed the low level of myosin 1C in the tumor compared to the corresponding mucosa as well. And this is a selection of some samples from the different stage, from stage 3 to stage 4. So it means that it has a correlation between the mRNA level and the protein level in down regulation of myosin 1C. So the Western blot result, when we analyze the members of PI3 kinase and BRA signaling, we found out that P10, ERK, and P-ERK protein were significantly lower in tumor samples compared to corresponding mucosa. This quite come to our surprise, but we may think at the beginning that things happen when the cells are nearby, and then it's starting to behave like a cancer. So the conclusion of the paper 3, myosin 1C was significantly downregulated in colorectal cancer, but this downregulation is not correlated to the tumor stage. And there's no link between low level of myosin 1C and activation of neither PI3 kinase or RAS signaling. This is a little bit not the same result that we discovered from the second paper. However, this may say that earlier identified correlation between myosin 1C in vitro that we test in the cells is not reproduced, and this may suggest a short-term effect of myosin 1C on PI3 kinase. On the paper 4, knockdown of myosin 1C resulted in early activation of the PI3 kinase pathway and disruption of key genes and pathway involving in apoptosis and cell cycle progression. The aim of this paper is to investigate effect of myosin 1C knockdown in a high throughput genes expression to identify new genes and pathway that it is involved. Because from the previous paper we're saying that it is involved in the PI3 kinase and ACT signaling. However, we cannot say that it is exactly or just only PI3 kinase. So we try to investigate more if it is involved in other pathway or there's another genes that involved. So the experiment we perform genes knockdown using siRNA in the time series format, and then we collect the samples from after the transfection from 0 up to 72 hours. And then we select RNA, 10 pairs of RNA that containing a pair of cells that transfected with siRNA and the cell that transfected with scrambled siRNA. So we select a 10 time point and subject to the gene expression microarray. So when we collect the expression data, what we did, we used the bioinformatics tools to analyze this data. First, we have to do the global normalization. So the result in this context, we did not use the housekeeping genes or we did not use any genes to normalize the data, but we used the things called globally normalization. It means that we use all the genes to normalize each other. And then we group the genes using the program called DRUM, Dynamics Regulator Event Miners. So to group the genes that behave in the same pattern. And then we analyze each pattern in I'm sorry. And then we analyze each one using the David and click pathway as well. So the first result, we found out that there is an early and significant response of knockdown of myosin 1C in PI3 kinase and ACT signaling. So we found out that 6 genes from this pathway show expression pattern highly correlated to the one that to that of myosin 1C. And in a late response, we found out the genes and pathway with known cancer-related function were identified. So in the late response, we found out that the pathway that involved in the prostate cancer, endometrium cancer, colorectal cancer are identified at the different time points. So the conclusion of this paper, we find that, or we identify that we find early response of PI3 kinase to the myosin 1C downregulation, and this support the paper too and explain the lack of correlation between myosin 1C and expression and activation of the component of PI3 kinase in the colorectal samples. However, we have to identify new genes and pathway for the further investigation later on. So the concluding remarks of this thesis, through this work we could provide evidence of tumor suppressor activities of myosin 1C through the involvement in cell proliferation, cell adhesion, from cells contact inhibition, and then fast induction of PI3 kinase and ACT signaling. Second, myosin 1C is significantly downregulated in endometrium cancer and colorectal cancer. And third, we identify new pathway through which myosin 1C may exert its function. So, for the future perspective, the data from microarray need to be verified by using the qPCR for the certain genes and pathway, direct identification of protein-protein interaction through the pull-down experiment, and this has been going on project in our research group using the yeast-2 hybrid. And last, we have to investigate myosin 1C expression in other type of cancer in order to say that it is generalized. This approach together will certainly produce further evidence for the functional significance of myosin 1C in cancer. However, lastly, IMPP5K is also the second candidate that was identified in the first paper, also need to be attended. Taking all into consideration, identification of novel tumor suppressor genes may not only help to further understand cancer pathogenesis, but it also gives a hint of hope to the new treatment to fight with the cancers. So I'm going to end my thesis presentation by acknowledging my supervisor, Professor Afros Bebudi, Professor Katerina Eyekwer, Professor Anders Olfusch, and Professor Colin Kringa-Levon, research group and collaboration Beyond Ocean, Anna Pfister, Neha Singh, Eva Falk, Imon van den Bolt, who is our collaboration in University of Pretoria in South Africa, colleagues at the University of Curve, Angelica, Sanya, Benjamin, Kalilia Kaisa, Lisa, Jessica, and Jasmine, and the colleague at the clinical genetics, Professor Tommy, who is not here, Rosemary, Susan, Naili, Anna, Morlin, and I cannot have today if I don't have the help from Elizabeth and Ingrid. And thank you. And before that, this work has been supported by Åke Fonden, Lundgren Foundation, Swedish Medical Association, also Nielsen Haley donations, and University Hospital Solgrenska. Thank you very much.
So now it's time for the question.
So, Kitty, let me start by congratulating you on the achievements during your thesis, for your nice presentation. You've elaborated on your findings of this